From Microbe TV, this is TWiEVO, This Week in Evolution, episode 49, recorded on November 27th, 2019. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels, Elde.
Hey, Vincent. Good to be back on the airwaves from Elde Lab Studios.
I saw you last week, man.
Yeah, in kind of an exotic location. We were at the giant virus meeting near Tegernsee, Germany.
And also with us, and we did a TWiV there, you and me, and he is joining us today from Austin, Texas, Rich Condit.
Hi there, Nels. Hi, Vincent. How are you?
Have you been on TWiEVO before?
Yes, once. We did some sort of epoxy thing. I forget what it was.
Correct. Welcome back, Rich. So I've got it here.
Thank you.
TWiEVO 21, a virus with a green thumb.
Oh, wow. I remember that. Yeah.
What was that about?
So this is a really cool paper out of Derek Walsh's crew.
Oh, okay.
Stern. And so the idea was that vaccinia the model pox virus, has ways of customizing, so to speak, the ribosome to favor the translation of virus messenger RNAs over the host. And it's this kind of unusual stretch of poly A, but not at the 3' side of the gene, which everyone's familiar with for messenger RNA, but at the 5' untranslated region that helps the virus. Yep. So we talked, I remember that was a really fun episode. We talked about the old days with Harry Noller. We even, I think there's a photo or two that we threw around from back in the day. Yeah.
Yeah, that's great. And that's a fascinating phenomenon. Well, one of the cool things about it was the poly A heads were discovered, I don't know, 20 or 30 years before this paper. And it's one of these things where people were wondering forever, does this really do something, or is it just kind of there? All right. And it works out that it was probably selected. It does something. Very cool.
Yep. The answer was yes. And it even extended to some plant genomes. Anyway, go back to episode 21, a virus with the green thumb. And to our listeners, it's worth it to revisit. Cool paper.
Say, wait a minute. Do you guys do the weather on TWiEVO?
Sometimes we do. Yeah.
Yeah, it kind of depends.
Because it's 56 degrees in Austin, partly cloudy. And this is as warm as it's going to get today.
Oh, wow. Yeah.
It's 13 Celsius and really cloudy here. There's a little notice. It says it's unhealthy for sensitive groups. So welcome. I'm sorry. What is it there, Nels?
I'll weigh in on the weather here. It's actually pretty consequential at the moment. We've got a blizzard pulling in to Salt Lake right now.
Oh, you got the bomb cyclone or whatever that is, right?
Right. So it's the first wave off of that. The first major moisture band arrived here just about an hour ago. And they're talking about, it's all over, the forecasts are all over the map, but there is some idea that there could be 4 to 5 feet in the mountains. So plenty of people around here are kind of salivating at the opening of ski season.
Yeah, right. Cool. And how far, are you going to get snowed into the LD studio?
We're looking pretty good at the moment. So it might be half a foot to a foot. And, this isn't our first rodeo. So we'll figure out how to navigate for the Thanksgiving holiday.
Yeah, you can walk home, can't you? You're not too far away.
No, that works. Exactly. And maybe more importantly, so a lot of the folks in my lab who are traveling decided, for example, to get out last night instead of today, which I think was a wise move. And so the campus is pretty quiet today as people are adjusting their plans forward to avoid the big dump here on the way.
Yeah, I used to love that on the rare occasions that I was around in the at work and everybody else was gone, and I felt, it's just a special feeling. It's nice.
Yep.
Well, anyway, welcome back, Rich, to TWiEVO.
Thank you. Glad to be here.
We thought, all of us last week were in Germany at the giant virus biology meeting, the fourth one in Tegernsee. And we thought we needed to do a TWiEVO for November. So we thought, why don't we just talk about the meeting?
And for those who, really don't want to bother looking at Google Maps, Tegernsee is in Bavaria, about, an hour train ride south of Munich.
Yeah. Yep. I'm just looking at the pictures. This morning, Matthias sent a link to some pictures.
Oh, yeah.
And I just pasted one into the show notes.
Yeah.
There's Nels with his giant virus drawing.
Look at that. Yep. So Matthias, by the way, we're talking about Matthias Fischer, who organized the giant virus meeting, and I thought he ran it beautifully.
Oh, it's great. Yeah. He's amazing.
And this is the fourth meeting.
It's right.
It happened about every 2 years, and it was Matthias's brainchild from the get-go, and he's run every one. And it's that's a significant effort, but he loves the viruses. He loves getting people together. And I was thinking about this after this meeting. It reminds me of the early days of the poxvirus workshops. The first of those was in 1977. And I went to every one, and I was able to witness the effect of those meetings on the field. Mm-hmm. And it's remarkable because you get people together and there's an exchange of ideas. But I think as much as anything else, this sort of, it energizes people and it energizes the field. And also, there'll be several really good papers that sort of set a standard that people aspire to, and it really gets things going. So organizing and running a meeting like this is really a very significant contribution to the field, and I think Matthias has done a great job with this.
I was very lucky to be at the first one where I did a TWiEVO, and we were asked to go back at every one, but couldn't make it for various reasons. The cool thing about this meeting, first, there are only 50 people, which is great because that way you can just talk to everybody. You're not lost in a crowd. Mm-hmm. And the second part is the venue, which is just fabulous. Yeah. So this is a castle owned by the Max Planck Institute. They bought it from the person who built it.
He donated it.
He donated it? Really? Okay. Yeah. I didn't have to buy it.
The Duke had no heirs, and a significant fortune, including his castle. And he looked around for somebody to give it to and tried out several different ideas that for one reason or another didn't really work, and then decided he would give his inheritance, at least to start with, the castle, to science, and approached the Max Planck Institute. And they weren't stupid. They said, yeah, sure, we'd love to have this castle, but the upkeep is we can't do it. So you need to give us everything else too.
Is that right?
And I don't know if he gave him everything else, but it's a significant endowment, enough to keep the place rolling.
Yeah, it's not cheap. But they also run 2 meetings a week, and they get money for that, right?
But we heard about that on the tour, and I couldn't help thinking, man, I'd love to close a deal like that where I get a cash and the cash.
Yeah, for sure.
Pay for real estate, and that's a pretty good. You actually get paid to take on real estate. That's a novel way of doing things.
Yeah, it is not too old as castles go, though, right? It's like just 100 years old or so.
Yeah, correct. So it started in the 1920s, I think. Is that right?
Sounds right.
Sounds about right. And it's up on the hill overlooking this little town of Tegernsee. There's a lake and it's apparently a resort. Lots of hotels and looks like apartments lining the hills. We 3 of us walked around, what was it, Sunday, right? Mm-hmm. Had some nice Greek food for lunch. Of course, you go to Germany, what do? You have Greek food, right?
Yeah, kind of a small resort town, really posh. That lake is just beautiful, and the surroundings also just incredible. Great place to have a meeting.
Yeah, it's just fabulous. So you're kind of up a hill. I guess you could walk down to town, but it would take you a while, right? Mm-hmm. So most people stuck around. I didn't leave the castle the whole time.
Neither did I. Yeah.
But I think that's great for having people just talk to each other. The food is really good. The rooms are amazing. They're like all wood with huge beds and huge tables and this big ceramic thing that I guess used to be a heater at one time.
Yeah.
Right?
That's right. Yeah. Well, and I heard, so Vincent, you were meant to be staying in the Duke's bedroom.
Yeah, I told them I didn't want to stay in there.
Yeah, so I got slotted in there, which was
because I knew they run a tour through it, you know.
We did the tour through it. There's also a massive bathtub in no shower. So I think that was the first time I took consecutive baths.
Is that in the middle of the room, or is it got its own little the tub?
Tub is in the bathroom corner, but it's a long tub, so you can actually soak pretty comfortably in there, as I learned over the course of the 3 days or 4 days of the meeting.
Food was good too, right?
Food was good.
That's great.
Yeah, we started with that Greek food, but then went into much more traditional Bavarian fare.
Yeah.
Great dining room, big, massive fireplace, high ceilings, and just really easy to kind of sit down next to different people every time we had a meal and just strike up a conversation. It's kind of really well done as a meeting.
It's great. It's really one of the better meetings. And I mean, I'll put some pictures of the venue in the show notes, the courtesy of Matthias, because he took some nice photos of us eating dinner, and he's got some really nice close-ups of everyone that really show people thinking or smiling. Really good.
Just to echo what Rich said, I mean, putting together a meeting, even a relatively small meeting, is a tremendous amount of work.
Yeah.
And so I agree 100% that contribution, especially for a field that's kind of figuring out what it is and who's involved in it. That's really pivotal. And so I think Matthias is really one of the best you could hope for to sort of be involved at that level.
And the cost for this is not really that much. I mean, I think we were all paid for, but I think it was 380 euros for
something like that. Yeah.
Staying there and 3 meals, you know, and
yeah, that's nothing.
Nothing. It's nothing. So I suppose that Max Planck is subsidizing it, but
yeah, probably.
So we thank the Max Planck.
Yeah.
Very much.
Great.
Now, the three of us did a TWiEV there. That's TWiEV 575. You can check that out over at microbe.tv with 3 guests, Alex Warden, Steve Wilhelm, and Asaf Vardy. Great TWiEV. We all had a great time, right?
Yep.
Totally. Yep. That was good. The kind of the theme of ocean ecology. I mean, that's one of the fun things about this meeting, I thought, was sort of the confluence of virology, of course. We're talking about giant viruses, but then with the ocean ecology. So you find these giant, we'll be talking in more detail about these giant viruses in particular, but these things are enriched in the ocean. So you have ecologists who, you know, either are interested in the microbial life in the ocean already, and this as a field is really taking off. You also have a huge input of structural biology, evolution, as we'll be kind of focusing on, of course, for the TWiEVO today, and then also genome science, metagenomics, and how that sort of, all these things are sort of swirling together and the kind of new advances that might be possible. And so, yeah, no, it was really something.
So as a result of being at this meeting and hearing so much ecology swirling around viruses, I decided to add an ecology lecture to my virology course.
Yeah.
Which I think makes perfect sense because you talk about how viruses impact the environment, right? Like the best example is EHUX, right? Emiliana Huxley makes these huge blooms in the ocean, and then viruses infect them and the blooms go away. I mean, what better ecological story than that? But there are many, many more. And then, of course, you can even talk about the ecology of, you know, mosquitoes delivering viruses and the ecology of infections within an organism. So I think I got inspired to do an ecology lecture. So that's pretty cool. I'm excited to make a new lecture this year.
That's great. You know, maybe historically or traditionally, because viruses are so small, we wouldn't think of the impact being on a logical scale. But that comes out beautifully in that TWiEV episode as we hear about the brown tides, as we hear about EHUX. Yep. It's crazy algal cell.
And one thing that Steve Wilhelm said, every year viruses release 150 gigatons of carbon in the oceans.
Yeah, they're really, really important in the carbon cycle, which of course is, critical to us, always has been, but is of special importance for us now in the era of climate change.
Absolutely.
And we didn't even know that, 15 years ago, we had no clue that they call it the viral shunt. We thought, okay, there's stuff in the ocean, things eat it, and that's how it decomposes. But now we understand that viruses do this. It's just awesome.
I agree. And this is kind of one of the themes for TWiEVO is that, even in the last generation or two of science, that things were really pulled apart. So virologists might be thinking really carefully, kind of in the molecular biology world, right? How do I even find out the DNA sequence? And that could be an investment of years, as I know of you have put in those kind of investments in your scientific careers. And so to be thinking all the way on that other end of 150 gigatons of carbon turnover in the virus shunt, I mean, this is what I think is really fun to see these fields sort of collide in real time now, and to have these conversations together in some ways for the first time. It feels a little overdue, but it's so fun to see it happening.
Yep. So actually, we decided we had such a good time together, we just wanted to extend it with another podcast.
That's right. So this is our meeting wrap-up, where we'll kind of cover our favorite talks and go into a little bit of detail about the stuff that really kind of caught our eye, and try to share that with our listeners today.
I have notes here if you need any fill-ins. I took a bunch of notes, as Rich will attest, because I sat next to him.
That's
great. He has his little red book.
I have a red book. I write with a pen. For years I used to take notes on my laptop, and I don't know. I kind of like writing. So I've gone back to writing in a little book, and I can carry it around. It's kind of, cool. It's just easier than a laptop. I don't know.
Yeah.
But before we start, I want to just briefly talk about this term that we talked about in TWiV, but the TWiEVO listeners should hear it too. NCLDV, which was mentioned many, many times. Nucleocytoplasmic large DNA-containing viruses. So all these viruses at this meeting are NCLDVs, right?
Right.
But that's not a formal taxonomic designation. It's just kind of loose association of all these viruses, right?
Yeah, the group, and I'm not really up to speed on this, Nels may know more, but the group that comprises what we now think of as NCLDVs at various times has been proposed as a taxon. And the most recent that I'm familiar with was the proposal that it be an order called Megavirales.
I see.
But I'm not sure that I don't know whether that's gaining any traction. Do you know anything, Nels?
Yeah, not a whole lot more, except that I would say the NCLDV, that sort of bulky, it kind of actually fits because we're talking about these bulky viruses in terms of their genomes and everything. But the sort of bulky acronym is a little bit of a working definition. So there's certainly, these large double-strand DNA viruses, and almost every month we're learning more about the diversity and just glimpsing. It's almost the tip of the iceberg kind of analogy here. And so how do these things all relate? Rich actually talked about this. We'll get into a little bit of that. Is so this set of core genes that might sort of infer that there's a common ancestor here somehow. But these are viruses. And so it's that notion of vertical inheritance sort of from generation to generation. We can throw that out the window pretty quickly. And then given enough time, the evolutionary flexibility here seems like it's pretty vast. And so how does NCLDV really hold together taxonomically? I think that's still being litigated as people are discovering new viruses. And this is kind of a fun part of the conversation as well. Is so pox viruses that both Rich and my lab have been working on, do they belong? And that was really, I thought, a fun kind of part of sort of side part of the meeting in a sense is should pox viruses even be at the table at a giant virus?
It becomes a real, for me at least, I don't know how other people feel about this, but a really sort of shocking question. Okay, because I have always thought of pox viruses as the original granddaddy of the NCLDVs. When I first started, because people have been working on pox viruses ever since we knew anything about viruses and before, right? Because the first is smallpox with Jenner over 200 years ago. I used to introduce my lectures by saying pox viruses are the largest viruses known. And now, among the NCLDVs, they are the smallest. And they also, I think in the beginning, when it was recognized that there was this apparent association amongst these, or at least that some of these other viruses were DNA viruses that had at least part of their replication cycle in the cytoplasm, all right? I always thought of the pox viruses as sort of the model system for all of this. And now as this is sort of growing, as we'll discuss, questions come up as to whether pox viruses even belong there. Mm-hmm. By the way, I just looked up a quick bit of history here. This is a fairly recent field. The first giant virus was discovered in 2003.
Yep.
So that's just 16 years ago. And it kind of ambled along for a while with a couple of groups digging up a few of these viruses over time. And now, to me, it just looks like it's exploded. And it's clear that these things are everywhere.
Yeah, totally agree. So that those early days, early 2000s, this was French groups that were looking in water cooler towers for some reason, fresh water. And the real insight here, the breakthrough, was that when people are doing virus discovery, basically the first thing you want to do is get rid of all the other stuff or try to enrich for the virus particles so that you can look at the DNA, so that you can look at the genomes. And so often what you do then is put them over a 0.2 micron filter to filter out or trap all of the bacteria and let the viruses go through. Now, if you're a giant virus, you're the size of bacteria or maybe even bigger, as we're now discovering in recent years. And so you're basically throwing away a massive part of the virus diversity that you're potentially interested in. And so that was the real breakthrough when, the sequencing technology got good enough that you could just sequence in bulk, basically, without doing that filtering. You have a trade-off that you're throwing away all kinds of other microbial genomes, and that makes it much more complex a mixture to sort of interpret. But that's where these giant viruses came from. So they were bigger than things that were seen before. The pox viruses we knew about because of that close human association with smallpox, stretching back hundreds of years. But all of this stuff sort of hanging out, whether it's in a puddle, the ocean, a water cooler tower, was just sort of hiding right under our noses. And so that's really kind of set off this whole thing.
Now people stumble into them, right? Like Alex Warden, she's looking at choanos in the ocean and finds a giant virus associated with it, right?
Yeah, no, and that was a fun collision actually between recent TWiEVO, so our last episode, thinking about this new choanoflagellate that Nicole King's lab discovered. Turned out they're hanging out on the island of Curacao together.
Yeah.
And found some really cool stuff together, a cool scientific collision. And so you can hear all about that on that TWiEV episode that we recorded with Alex. She really got into the weeds with us on the backstory. It was really fun to hear some of those details. And
like we were talking about earlier, this is a convergence of different fields. Alex is not a virologist, right?
No, she's an ocean ecologist. Right?
She's an ocean ecologist who just ran into this virus.
Yeah, and one of the things that's fascinating here is because these viruses, not surprisingly, are giant in the sense that they're big and they have big genomes, they start encoding really cool things. And one of the really surprises from her work as they were putting together the genome of these choanoflagellate viruses was that they encode some of the rhodopsin pathway. And again, sort of echoing on that TWiEVO, episode 48, where Nicole King's group discovered how there's an interesting developmental switch in that host, the choanoflagellate host, using the rhodopsin pathway. And so it's all this biological complexity happening that's just kind of coming under our radar for the very first time.
So the idea go ahead.
For the TWiEV audience out there, when you hear rhodopsin, one of the things you can think is vision. Rhodopsin is the molecule that is in our retina and allows us to interpret light hitting our eyes as images. It's vision. But in a more general sense, correct me if I've got this wrong, but rhodopsin is a way to turn light energy into other kinds of practical biological energy.
You could, or you could respond to it, right? So it's like with the choano, the cup choano that we did on the last TWiEVO. The light changes the way the organism is organized, goes from a feeding to a moving around form.
Right.
And that's in response to light. Right. Now, yeah, or it could make energy, sure. Now, this one, she found this virus in a unicellular choano. So it's not going to be, probably not going to be making such big differences like with the cup-shaped choano. But I don't think we actually even speculated during the TWiEVO what it might be doing, right?
No, exactly. So why would a virus encode these genes? So these things are generally, that's part of what she shared with us is, first of all, where did these genes come from? Did the virus evolve them de novo? Probably not. They look like they're distantly related to some rhodopsins that you see in bacteria. That probably means that it didn't get it from its choanoflagellate host, but through sort of a third association, potentially. One of the major themes that I would say emerged from the giant virus meeting is that these viruses are gene thieves. They're just sampling genes from all around their hosts, all around the genetic associations with other, or just not even just biological associations with bacteria, with archaea. And so you end up with these virus genomes that are sort of this mosaic or kind of chimeric grab bag of genes from all over the place. And then the question becomes, is this, from an evolutionary standpoint, is natural selection acting on this? So is there some advantage that the virus gains or the virus-infected cell gains by having these things around? Or so I think on first principles, we might say that it's an accident that the virus gained these genes in the first place. But then the question becomes, after these things are being sampled by almost countless numbers of virus particles or virus genomes out there, how does natural selection act on that? And does more complicated biology emerge from that?
I would guess, or one possibility is that, so these choanos are living in what she called sunlit waters, right? The ocean. So maybe the virus infects its host, it sets up these new rhodopsins, and now the host can make more energy for virus reproduction, right?
So one of the really tantalizing things about this particular choanovirus is that it encodes not only the rhodopsin, which is a protein, but in order for the rhodopsin to do its job, it needs a pigment, which is the actual molecule that senses the light, which is retinal, and that has to be fabricated using a biosynthetic pathway, a series of enzymes that take smaller molecules and hook them together properly to make this pigment. And the virus encodes not only the rhodopsin, but almost the entire pathway. I say almost because the first couple of steps are encoded in the choanoflagellate, and then the virus fills in all the rest of the steps to make this pigment. Now, if there's not selection going on there, I would be very surprised. It's actually interesting to think what came first. They didn't pick up all of this stuff at once. Yeah. Right? I mean, it's fascinating.
Yeah. And it also echoes, we haven't talked a little bit about this, but the bacterial on TWiEVO, the bacterial symbionts of insects, for example, where you start as people are sequencing those genomes and putting together the metabolic pathways, like the one that would lead to the creation of retinol, you start to see those patchworks of, the host encodes this, the bacteria encodes this, or in this case, the virus. And so it has that flavor of sort of almost a symbiotic relationship.
Yeah.
I want to go back to your discussion just now, Nels. You're talking about these giant viruses being thieves. And one of the cool things is that many of them are bigger than their genome. They have extra space, and you can stuff extra DNA in. And so that makes it easier to acquire DNAs. And I was thinking, not all viruses are thieves. Not anymore. They were thieves at one time, but polio hasn't gotten a new gene in ages, right? And why is that? So I was sitting at lunch with Nels, and I think it was Rich and Eugene Koonin. Yep. We had a good discussion about this. So some viruses are just too small. Polio, you can't fit more than 5% extra nucleic acid in the capsid, right? So they're stuck. but apparently that's okay. It works. Others aren't able to incorporate nucleic acid because they don't recombine. There's some RNA viruses that do not undergo recombination.
Yeah.
Because the way the polymerase is set up. And so that limits it out as well. So it's cool when you start to think, so you need room in the capsid, you need to have recombination in order to do this. And so that was a fun conversation. Were you next to me, Rich? I think you were.
I think I was there. Pox viruses, it's been known for some time, you can add, I think, up to about 25% of the starting size of the genome. You can increase it by about 25%, and you're okay. And since you're starting with 200 kilobases, that's another 50 kb. That's a lot of information. And some of the giant viruses, I would assume, have even more room than that.
Yes, yeah. For sure.
And that kind of marks, I think, one of the fundamental differences. So the reason that I think NCLDV, as bulky and sort of confusing as the definition might be, but all of these viruses are kind of have that replication style or that kind of organization where they are almost sort of tuned, it appears, to be able to do all of this kind of genetic sampling from the environment. Whereas I think on the other side of the divide are generally the RNA viruses like you're describing, Vincent, that are much more limited in their capacity to be gene thieves. Although there are some, I'd say, very notable exceptions.
Mm-hmm.
So,
sure.
For example, the retrovirus Rouse sarcoma virus, this virus that infects chickens, which was now 30, 40 years ago, a really confusing at the time finding of, hey, wait a minute, why is there a kinase gene from the host encoded in that retrovirus genome? This turned out to kind of be one of the fundamental clues about the presence of oncogenes in virus genomes or transforming genomes that led to ultimately to the Bishop Varmus Nobel Prize.
And there, of course, the reproduction cycle of the virus has a stage where the genome is integrated, and so perfect scenario to acquire some genes, right?
Mm-hmm.
Now, I mentioned the polio, the capsid being constrained. And I talked about this with Koonin. What about vesicular stomatitis virus? You can add a lot of extra sequence because the capsid the particle just gets longer. It's bullet shaped. You just make a longer bullet. Yeah. But so why doesn't VSV acquire genes? Because it doesn't recombine. Yeah. Never get recombination. So there you go. While we're on this, Nels, it makes sense to discuss your talk briefly because you talked about acquiring genes. You've made a model system where you force a pox virus to acquire a gene.
Yeah. Right. Yeah. So no, this idea of horizontal transfer as kind of a mechanism of mutation or with the potential for kind of really drastic evolution in just single steps, right? So with viruses, we constantly think about the point mutation, which can drive a lot of RNA virus evolution. Of course, there's also reassortment, there's recombination in some virus classes, not in others, as you're pointing out. But so we got really excited a few years ago about the fact that for pox viruses, where you have a ton of experimental tools available, a lot of them pioneered by Rich and that whole crew that was coming up studying the pox viruses, to take advantage of that, to try to set up systems to actually model that process of how do these things become gene thieves. And so we've been working on this for several years, 4 or 5 years now. And we kind of think we caught them red-handed. We caught them in the act. The viruses acquiring a host gene based on an experimental scheme we set up. And so the surprise was, so we knew going into those experiments that you needed a source of reverse transcriptase, something that could take host DNA, a host gene that is, and when we're talking about mammalian hosts, which is where vaccinia virus, which is where smallpox, et cetera, have been well studied. You've got massive genes just littered with introns. And yet, when you look at the virus genomes, they're all missing introns. They look like messages. And so what that gives you a strong hint that there is the action of reverse transcription going from a RNA intermediate lacking introns back to DNA. And so reverse transcriptases, you could probably think about two major sources. One are the retroviruses, which do that as part of their integration and their replication cycle. The other source that's common to these host cells are from retrotransposons. So these are the selfish genetic elements. We've talked about these on TWiEVO. And this kind of harkens back to Richard Dawkins' The Selfish Gene, where you have these little genomic parasites that just copy and paste. And so the fun discovery with our project was that it looks like those retrotransposons are playing a key role in facilitating the transfer of a host gene to a virus-infected cell. And in fact, when the retrotransposon makes a mistake, that is, it grabs a host message, mRNA, instead of its own message RNA to do the work of a selfish genetic element just over-replicating relative to Mendelian traits, you end up with a host transcript that could be either copied back to the host genome. There's thousands of these kind of littered through our genomes, the so-called processed pseudogenes. But now imagine in the infected cell with a large genome, like a pox virus, like a lot of the other NCLDVs, that could be a genomic target, and a processed pseudogene from the host could end up there. And we think that's one of the major mechanisms of how this works. And so that was the story I was telling at the meeting.
So I asked you a question. How often does this happen? Is it every time a new virus particle is replicating? Could it happen at every reproduction cycle? And you had some thoughts about that, right?
I do. Yeah, no, it's a great question. And so I think, first of all, it's probably happening more often than we think. There's actually some great evidence of this in insect viruses, the so-called baculoviruses, which are at least loosely affiliated among the NCLDVs, the giant viruses. And if this is some work from Clément Gilbert's lab, he's in France, and he found that actually up to 5% of the genomes that they collected from the baculoviruses could be interrupted by a transposon. So it's a DNA selfish genetic element. So the biology is a little different, but I think the theme is the same, which is that this could be happening all of the time. And when we think about, as we've talked in many episodes about sort of mutational mechanisms, whether it's a point mutation or any kind of mutational flavor, that most of these are probably going to be deleterious. You're going to insult a genome, in this case with an entire transposable element. Some of them might be neutral. You might land in a spot in the genome or be integrated in a spot in the genome where the virus can still replicate one way or another. It doesn't really influence it. Or in these rare cases, it might be beneficial. And so when we just look at these snapshots of viruses' genomes today, it's probably a collection of those really rare events that have just accumulated to sort of contribute to modern virology, to modern virus replication. But yeah, I think we're just looking at sort of and trying to understand what could be a really big force in virus evolution.
So this could be happening all the time, but only much more rarely shows up in the finished product, right?
That's right.
So how would you investigate that? Some sort of basically sequencing on infected cells in a fashion that could be sensitive to these rare events that might not wind up in virus particles. Is that a practical goal?
Yeah, it's a practical goal, but we have a technical hurdle. So that's exactly what we've been trying to do for the last couple of years, actually. And it just comes down to currently an obstacle of doing deep sequencing. And whether you're doing long reads or short reads, what we're looking for in the situation that you're describing, Rich, is for, like you're saying, a rare event. And so when we're sequencing a genome, for example, whether it's a virus, a bacteria, a eukaryote, whatever, one of the advantages there is we're usually starting with a homogeneous population, right? We might say, let's sequence this algae genome, EHUX or something like that. And so what you would do to begin is you'd say, let's get this population to be as identical as possible. Let's take a clone, a single cell. Let's allow it to divide a bunch of times so that we can get enough genomes to then do the sequencing. So that's great because then you can sequence to incredible depth and you can compare overlapping reads to discern whether what you're looking at is real or if it's an artifact of the sequencing process. Remember, these sequencers make errors. You have to build genomic libraries. Enzymes are at work. You put things together in ways that aren't actually represented in the genome itself. You have all these artifacts. So what's different about this problem or question is that you don't have the luxury of a homogeneous population. Everything is a little bit different. And in fact, you want to try to measure those rare events, that single guy, right? And so you can see them, and we can see thousands of them, but what we can't discern yet is whether what we're looking at is real or if it's an artifact. because we only have one read from this sort of diverse population. That turns out to be a pretty big problem for genomics at the moment. But I think one that's...
And it's like you can't go back and check and make sure if it's still there or something, because it's like, what you're looking for is something that might just happen transiently on a rare occasion. So you can't go back and say, is that reproducible? You don't expect it to be, really.
That's exactly right. So you can't go back and do PCR or something like this. And just as you're saying, most of these things aren't going to replicate. They had their one chance. Selected out.
I often think about the fact that in a pox virus factory in the cytoplasm, which is just chock full of DNA, or even in my mind, to a large extent, made of DNA. They stain with a DNA staining dye, DAPI. Okay, there's only a small percentage of that stuff that winds up in virus particles, and there's a very large percentage of what's in a factory that, I don't know. I mean, the virus doesn't care as long as it makes a couple of particles. Fine, that stuff just is part of the trash heap. But there's an enormous pool of information and stuff happening there, part of which is probably structural. In terms of the whole factory. Part of has to do with, maybe providing a scaffold for some of the assembly to take place and all the rest of that. But it's, in terms of sequence, it's to a very great extent unexplored.
Yeah, I agree. So as evolutionary geneticists, we're trash collectors. And and as we've discussed on TWiEVO before, with evolution, it's oftentimes the things you can't see that are kind of the most interesting in terms of understanding the evolutionary pathways or the principles. And it's those rare things that we can see, that where all of our focus is, just because it's a lot easier to study. But that's only a really small part of the bigger story here. Yeah. I
just have a little aside here that is striking me for the audience. All of us are looking at this document that we put together for this show, where we put down a sort of our favorites that we want to discuss or something like that. And I don't think we've gotten past talk briefly about NCLDV. This is like a dinner conversation. This is what happens at these meetings.
Oh, that's right. That's the idea, right?
Yeah.
Because
we should do that. We should tackle. There's so much fun biology to share here. Why don't we go after it? And I think we've got you listed here first, Rich, with some great talks to queue up.
So and we can go back and we can alternate, go back and forth amongst favorites or something like that.
Yeah.
I just kind of went through the program and picked out the ones that really caught my attention. One was a talk. Do I have this right, that this talk about PBCV structure was Thomas Close?
Yeah.
Thomas Close, yep.
There's a lot of people involved in this. As a matter of fact, there's a paper that I linked to here. Near atomic structure of a giant virus. First author is Q Fang, and the last author is Michael Rossman. And by the way, as we discussed on TWiV, Michael Rossman died within the last year, correct?
Mm-hmm. Yeah,
that's right.
Famous structural biologist who's been responsible for a lot of these large structures, and we dedicated a portion of the meeting was dedicated to him. This has a lot of folks on it, including Thomas Close and Jim Van Etten. And it is a solution of the structure of one of the first, it wasn't actually called a giant virus when it was first described. It's a chlorella virus. We've had Van Etten on before. This is a virus that infects a blue-green algae that lives inside a paramecium. So Paramecium bursaria chlorella virus 1. It's been around for a long time and is now included in the NCLDVs. So these, this was a description of the structure of the capsid for this thing, which is no mean feat, because these things are huge and extraordinarily complex. This thing has 5,040 copies of a major capsid protein, 60 copies of a penton protein, that's at the peaks of the icosahedral shell. And what was really striking in particular to me about this talk, 1,800 minor capsid proteins, of which there are 13 different types. Mm-hmm. And you can actually look at this paper. I think anybody could look at the figures from the paper. You may even be able to look at the paper. But one of the figures that really struck me is this structure with the major capsid protein removed, mm-hmm. Yes. That winds up showing the positions of all of the minor capsid proteins colored to indicate their different types. And it's beautiful. It's like this, net or scaffold of proteins that weave in and out of the major capsid protein to contribute to the overall structure, which makes perfect sense to me. In a structure this big and this complex, you might need some help from some minor structural proteins to keep the thing together. And it's extraordinarily complex. It's a feat to figure this out. And the structure is beautiful. One of the proteins that really blew my mind, and I don't, let me see, it's this dark or light blue guy here. It doesn't matter. Is this sort of tape measure protein.
Yep, that's what he called it.
that starts at a vertex or near a vertex. I think it actually loops around. No, it doesn't quite loop around a penton. It comes close, mm-hmm. But then weaves its way down the edge of one of these large triangular faces all the way to near the other vertex, mm-hmm, mm-hmm. as a sort of a glue. And there's 3 of them per vertex that sort of surround, 3 of them per triangular face that sort of surround this thing. And there was some comment along the line that these sorts of proteins now have been deciphered in a few different giant viruses, and they have different lengths depending on the size of the capsid, of course. Mm-hmm. Unbelievable.
Yeah, this feels to me like virology on steroids.
For sure.
Well, a lot of the giant viruses. This was I was elbowing Vincent during these talks. There was this and that on steroids throughout the meeting.
Yeah.
We're going to come to a plaque essay on steroids.
This, what I thought was cool about this talk is that there's a spine at one 5-fold axis on this virus, right? Which, because there's only one, if you apply icosahedral symmetry to your structure, you won't see it, mm-hmm. And so you have to take that away. You have to not apply, and this is something that people are doing more and more in structures now, not applying any symmetry and see what you get, because there are cases when you have like this, a spine at one axis. Sometimes when virus is attached to cells, you get different things happening at that one attachment site. And so this is a finger-like projection made up of a variety of proteins, which I thought was really cool. Probably involved in attachment, I would guess.
In fact, one of the on steroids comments happened during this talk. I elbowed Vincent. I don't think it was this talk. It was another one. Elbowed Vincent and said, "This is quasi equivalence on steroids."
Yeah, that's right. That's okay? Right. That was one of the last talks. Yeah,
right. Because the quasi equivalence refers to the fact that this is an old concept. Tell me if I get this wrong, okay? Because it's quite possible. But refers to the puzzle of how do you take the same protein and use it over and over to make an icosahedral structure where at some points in the icosahedron you have hexagonal symmetry or threefold symmetry, and other places you have 5-fold symmetry. In order to do that, you have to make the protein-protein contacts a little different in some places. So they're not quite the same. They're quasi-equivalent.
That's right.
All right?
That's right.
And in this particular structure, that has been worked out at the penton into, I guess, Vincent, you said that you guys, I must have been absent for this. You'd done this paper about the spiral growth of the spiral symmetry around the penton to make the capsid.
Yeah, this trisymmetron and pentasymmetron.
Yeah, right.
Yeah, we did it. I thought you were there because I think River, who is the senior author, that's his first name, which is kind of his.
He was also at the meeting and gave a nice tribute to Michael Rossman.
Yes,
sure. But let me give you his real name, because River is what he calls himself. His real name is Xuan Shao. Okay, Xuan Chuan, and then he calls himself River and XAIO. He's at the University of Texas El Paso, and he likes to wear cowboy hats, which is pretty funny because he's Chinese. So he solved that interesting, and that happened to be Cafeteria rhombogensis virus,
right?
Which he solved, which was huge. And I remember at the first giant virus meeting, he said he had a preliminary structure. He said, but there were so many atoms in it, it would continually crash his computer. And, this is now, 8 years ago. So computing has advanced since then. And he said every frame, he made a movie, and you can find it on his website, which is this virus rolling on the ocean, right? He said every frame took him 24 hours or something like that to render on the computer because there's so many atoms in it. But yes, he has this interesting way of assembling the particle in a spiral fashion, and we did talk about that. Does
that apply to PCBV or PBCV?
Yes, I believe it's similar. Right. PBCV, CROV, and I think also the anophageferens virus of Steve Wilhelm's is also a similar structure. Yeah.
Yeah, and I think all this complexity is what really draws structural biologists almost like moths to the flame, right? To try to
yeah, for sure.
Try to understand how these relatively simple structures that are pretty well described for smaller, simpler viruses, how you can elaborate on them, or even just kind of use those principles to then build these really complex things. And it's just at the perfect time because with cryo-EM, all the advances, as you're saying, in computing, and then bringing that together with crystal structures, you can now, for the first time, put together these massive things in really high resolution, and that's a pretty powerful place to be. So, can I ask you guys? So, for these icosahedral viruses, there's this T number that was often spoken about. These things are really off the charts, aren't they? In terms
of So the T number is the number, if the simplest way, the triangular face of an icosahedron, the number of facets in that face, and you can go start from T equals 1. T equals 3, polio, and you go up, and there's certain numbers according to the original theory of Casper and Klug that are okay, but we're seeing viruses that apparently violate those rules, and they're huge, hundreds and hundreds of T numbers now. Yeah, these viruses have a lot of segments per triangular face.
Yeah.
This TWiEVO 506, you were not with us, Rich, that's right. But there we talked about this structure, CROV, that River had solved, and this new.
I'm actually glad to hear I wasn't there because I don't remember it.
Yeah, you weren't there. That's right. And so I think he had sent us because he had just published it, and I always told him when you publish it, I want to know because we want to talk about it. We talked about it, and then he followed up with an email, which was really nice. So it's amazing. Part of the appeal, to find new principles, but also to see how big a structure you can solve. And now with cryo-EM, it's getting easier because you don't need to make crystals anymore, right?
Yeah, that's right. And these things are so big.
That's a big deal.
Yep. These things are so big that you can actually start to get that scaffold at reasonably high resolution. And then as you overlay some of the more traditional crystal structures, now these things are kind of springing to life in terms of the resolution. And you could I could imagine spending an entire career just sort of tooling around from layer to layer and just seeing, learning the rules of how you can put together these complex new things.
So the triangulation number of PBCV1 is 169.
It's small.
The triangulation numbers of Mimi virus, which are PBCV is 1900 angstroms. What's that? 2 microns. Mm-hmm. Is that right? 2 nano. No, it's 2. Yeah, it's 2 microns. Holy cow! Yeah. Mimi virus is 2.5 times that. It's 5,000, in diameter. Its triangulation number, depending on what you're talking about, is somewhere between 972 and 1,200.
So the biggest particles are about 1.5 microns. That's like the ones from Siberia. Pandora, the Pandora viruses. They're 1.5 microns, which is 1,500 angstroms. So these can't be that big. We're not talking about microns here.
I'm looking at a chart here from a paper that's has the capsid diameter in angstroms, 1,850 angstrom, or 1,900 for PBCV, and 5,000 angstroms for mimivirus.
So that's 5 nanometers.
5 nanometers,
which is not 5 microns. No,
yeah. Okay,
it's much smaller.
Yes.
I also, since we're on structure, Steve Wilhelm talked about the structure of his brown tide virus, which is anophageferens. Oreococcus anophageferens is the name of the algae that makes these brown tide blooms. And then he isolated a virus some time ago, which he calls AAV, unfortunately. He said they didn't call it brown tide virus, BTV, because they're afraid it would conflict with blue tongue virus. So instead it conflicts with adeno-associated virus. So you can't, don't worry about it.
Well, you can't win, yeah.
No.
Forget it.
So his structure is very much like PBCV1, which we just talked about. It has a unique vertex. And he showed a picture where, so these viruses are cool because underneath the capsid, they have a membrane inside the particle, right?
Yeah.
And in his, you can see this unique vertex with the projection, and under it, the membrane is kind of pulled away from it. Maybe getting ready to put the DNA into the cell, who knows? And the cool thing about his story is that a number of enzymes are packaged into the virus particle. They're polymerases, and they're polysaccharide degrading proteins. And the one he talked about was a pectate lyase. Mm-hmm. And he thinks maybe this is involved in getting into the host because, the host can have carbohydrates and so forth. But he said and this is another thing we can bring up, that there are data sets from cruises that people take around the world in different oceans, and they collect water, they sequence it, and they put that online. And there's one called the Tara Oceans data set, right? And he said you can look for your gene. You could look for pectate lyase, and you can see it's all over the place.
Yeah.
I thought that was very cool.
I agree. It's sort of another example of, it almost feels like these viruses are having a biological conversation with their hosts that's somehow recorded, or there are clues to it in the genomes or in the proteomes, as the virology is just starting to get, folks are starting to tackle it. And so that's another example, right, of all of this, when you have these complex systems colliding, where you can start to learn about these biological interactions in a new way. Yeah, that was a really fun story.
So just to follow up, the Tara Oceans data these are scientific expeditions that were done between 2009 and 2013, and they sample water basically, and they sequence it, and they publish it, and they go to different places. There's different oceans, polar circle, et cetera, et cetera. And the cool thing is that you can access these data and query it for whatever you want. Mm-hmm. Awesome stuff. Awesome. Yeah. So Nels, what did you like?
Yeah, so I thought I would jump in with a couple talks that were on different viruses, but sort of had a similar theme, which is actually, it almost feels like those Russian nesting dolls, where you have complexity, like you have a host, and then you have a virus, and then you have something within that, and something within that. You keep peeling back layers of the onion almost. So I'm thinking about the talks from Chantal Abrigel. And so she was telling us about some mimiviruses that infect Canthamoeba. So these are, single-celled protists that you can find kind of all over the place, both in freshwater and soil and in ocean environments. The mimiviruses, these giant viruses, and what she and others had, reported in not super recently, but in the last few years, are these ideas of virophages, so viruses that infect viruses. And the one that got some acclaim a few years ago, they called Sputnik. And so this is the system that she's been working on. And then the kind of new wrinkle on it is actually this idea that as you go kind of peel the next layer of the onion, there are these things called transpovirons. And so these now associate with the virophages. And you now have, 3 or 4 levels of kind of parasites on top of parasites, or as there's that old saying, fleas on fleas kind of idea. This is happening in the giant virus world in really kind of cool and interesting ways. And so then Matthias Fischer, who we already were sort of singing his praises for organizing such a great and fun meeting. But so he discovered this massive virus or giant virus in a algae species called Cafeteria. And so he was telling us about their recent efforts to actually sequence the host genomes of the algae themselves. And I think they have 4 strains now sequenced. It hasn't been published yet. And what was really kind of interesting about this is as they were scanning along those algal genomes, oftentimes what people do as they're just started doing genetic analysis is you'll get a sense for what's the GC percentage of the nucleotides. And that can range pretty wildly into, it might be 20% on the low end or 70% on the high end, depending on the genome that you're looking at. So for that algae, Cafeteria, which I think is named, the genus is named for how prolifically these things graze and eat when they're out in their aquatic environments. Yep. So most of that genome is 70% G and C, meaning that only 30% are A's and T's of the 4 nucleic acid bases. However, what they noticed as they're scanning along is regions of the genome that make a severe drop down to 30 or 50% GC. And it's almost like, this isn't just sort of random chance. These are long stretches where the genomic content really changes in terms of the base pair composition. So as they look more closely, they found these virophages. So this would be the first in that kind of layered onion analogy or nested doll analogy, sort of the third level as you go from host to virus of the virus. And so they call these things, they kind of are starting now to characterize because they have the whole genomes, the families of these virophages that they're naming emails based on, I forget how they came up with the analogy. It's pretty clever. And so you look at the emails, and they're just you're starting to characterize these biological emails, which are some remnant of past infections where the host genomes were first infected with the virus of Cafeteria, the giant virus, and then the virophages associated with that themselves have integrated into the genome. And then there's sort of this third wrinkle, echoing on Chantal Abrigel's talk, which there's these retro transpovirons in this case, or transpovirons. These, I don't know if these are retro or not, actually. That would depend on how they were inserted, but they're actually interrupting the email sequence. So you have this drop down to GC content that's a lot lower, and these little blips where it actually returns closer to the host genome, and this is that third layer. So it's almost like an archaeological dig in the genome for all of its complex biology. And the reason I love this is because now, as we're working over here in poxviruses, and we're thinking about, how do these transposons or these little snippets of DNA, these parasitic snippets of DNA, how might they be mobilizing genes between genomes? And I wondered, and Matthias and I talked over a beer or two, whether or not this is maybe how the Cafeteria virus that he does it. Maybe there's a story in here about how these things are moving genetic material as they're inserting in and out of the host genome. And so,
gotta be. I
think so. He was pretty cautious at this point. So right now he has some sequences, and they're working on it. But that's I think that's one of the actually bigger themes I wanted to bring up in our conversation, is that the sort of divide between the tools that we have in NCLDVs like poxviruses, based on those decades of work from the community of poxvirologists, which put us in this spectacular position to take sort of experimental approaches, versus the challenges of kind of this frontline giant virology where you're discovering new stuff. And so just glimpsing at those emails interrupted by transpovirons, that's the first time anyone's looked at it. And so Matthias and his lab and others are working kind of frantically to try to bring in some of these genetic tools. Can you actually do genetic manipulations, both of the host and of the viruses, in order to test some of these ideas? And so that's kind of the tension, I would say, between the fields, is that when you're just seeing something for the first time, and you might only have some sequence, let alone the virus, and let alone tools to study it. And so for me, at least, that's a fun part of the conversation, is to see whether we can use the experimental systems to learn some of the concepts, and then hopefully advance experimental tools in these fascinating, complex giant viruses as science moves forward, to sort of bring everything together into a more experimental focus.
I was talking with someone from Matthias's lab, and this is not easy because there are no plasmids, right? Which is a staple for what we do. They don't, you have to, if you want to use CRISPR-Cas to disrupt things, you have to get it in the nucleus, and they don't know how to do that yet.
Exactly.
So a lot of fundamental stuff has to be done.
Also, on the topic of tools, I had a conversation with Matthias over his poster, and the sequencing tools are actually kind of biased, because of their evolution, kind of biased against the discovery or sequencing of integrated virophages. And he talked about all of the, among other things, the GC content thing. He talked about all of the challenges to getting at these things by a sequencing approach. And the sort of the last straw was that after putting together one of these genome sequences with the virophages in it, they submitted it to GenBank at NCBI, and it was rejected with the note that their sequence is contaminated with viruses.
Great one.
Which is just a cool irony. I love it.
Yeah.
So significant challenges.
Yeah.
Now I forget so the virophages all encode integrases, so they're designed to do this.
That's right.
All right. The transpoviron, do those things make capsids, or are they just elements? I forget.
They're elements.
Yeah, they're elements.
No capsids. Yeah.
I agree. Right. Yeah. So then I thought another topic I wanted to bring up was actually the point that we've been touching on a little bit, and Rich mentioned, is do poxviruses belong with the NCLDVs? So I'd say, kind of picking up on the history of the field a little bit, there were those early 2000s discoveries. And then, as was already mentioned, it's been just sort of like a gold rush of giant viruses, where almost every month now there's a new record holder for, the size of the virus genome or the size of the virus particle, actually filling in all kinds of new branches, just entire new parts of the kind of giant virome are just starting, barely coming into focus. And what's crazy about some of these is that if you might put together, and the quality of the genome can vary, but you put together sort of a plausible hypothetical set of open reading frames or genes that might be encoded in that giant virus genome. You do a blast search or query other public databases, and it could be that more than 50% of the genes predicted are actually have never been seen before. I mean, that gives you a sense of like kind of the discovery or the frontiers aspects of the work. But what's starting to become more clear is that you've got this sort of growing collection in that giant virome that are all related to each other. And then when you now circle back and ask about the poxviruses, there's a dozen or so core genes, and I'll let Rich comment on this, since this is exactly what he talked about. But the relationship that's the sort of any proposed last common ancestor would be so ancient that there's been all of this time for virus evolution to kind of go in different directions. And so the question becomes, like, maybe from a functional standpoint, whether or not these things have diverged so much that is it even worth it as a model system for thinking about even the genes working. So that's kind of Rich picked up the story, I think, in a really interesting way.
Yeah, to complete your thought there, Nels, that was Morgan Gaia who was talking about phylogenetic trees and said that he got a much sort of tighter, more convincing phylogeny if he dropped the poxviruses out of it, right? Now we had a conversation later on with some people around dinner that where people mentioned, well, with any of these phylogenies, and you know more about this than I do, Nels, if you drop out a whole group, everything else tightens up, because every group that you add onto a big phylogeny is going to add some variety and make it a little fuzzier.
Yeah, that's right. So just to maybe comment on that briefly, and I'll let you go back to it, is that
Sure.
When you add more sequences from more diverged sources, when you're building this phylogenetic tree, you need to compare a character that's in common with everyone in that tree. And so if you add something that's diverged, there's just fewer shared characters across the entire comparison point. And so you lose a lot of data. And so the sort of confidence you have in the arrangement of the tree goes down a lot, the more greedy you get with trying to include more diversity. And so by dropping the poxviruses in this case, Morgan could get much higher confidence relationships between the other viruses that remained. So yeah.
Dude, that was that caused me a big sort of, face palm. Okay. Because, what? Wait a minute. We were here from the beginning. Now you're going to drop us out?
You remember that Fisher put up a picture? It said, "This tree is not to be trusted."
Yes, right. And he had like one of these road traffic caution signs with a big tree falling on somebody.
Yep.
That was great. Yeah.
So, when Matthias invited me to come to this meeting, my knee-jerk reaction was that I was, and this is, as I described in my talk, in retrospect, I think a certain level of naivete on my part. I knew from, kind of watching all of this stuff go by, that there was a common core of relatively conserved genes all throughout the NCLDVs. And this has been developed largely, I think, by Eugene Koonin. Though others as well, in particular, starting with the pox viruses, there was a common core of genes that are shared by all pox viruses. This reduces to, depending on who's counting and what you call conserved, anywhere between 20 and 40 genes. And frankly, this whole experience for me has kind of awakened me out of my retirement. This is an ongoing investigation for me. I have ideas for how to pursue this further. But cool. I was drawn in particular my knee-jerk reaction was, so there's this common core of genes, and at the same time, in the NCLDVs, because of some of the technical issues that we've already talked about, it's sometimes hard to get a real grip on the biology, and in particular the biochemistry or molecular biology of these viruses. And yet we know a lot because of all of the rich history and the technology to study pox viruses. We know a lot about the biology of pox viruses. So I can look through some of the major features that attract me to pox viruses and ask, and because of the conservation, I might be able to say, well, this is what the biology of pox viruses looks like. Since genes are conserved, you might anticipate that in these other viruses, these genes would be, or these processes might look similar. And so I chose the features of pox viruses that I'm most interested in, including some elements of structure, some elements of membrane biogenesis, some elements of transcription, where I knew that there were a couple of genes involved that were part of these core genes that were conserved. And actually, I enlisted the help of Eugene Koonin and Natalia Utin in putting together the talk because, for example, in the transcription stuff, I knew that one of the genes that I was most interested in was conserved throughout, but it's part of a larger process in the pox viruses that involves other genes. And I had them do the phylogeny for me on these other genes and found that they existed only in pox viruses and weren't conserved throughout. And the same held for all of these other processes, though, where I thought I might anticipate some conservation. In fact, they were only in the pox viruses. So this sort of reinforces the notion that the things that we think of as really signature processes in the pox viruses, like the complexity of the structure, the way the membrane is put together, some of the features of transcription, in fact, are highly evolved just within the pox viruses. And the ancestral pox virus probably, I didn't mention this, but it's fairly obvious, doesn't look anything like what we envision a pox virus will look like today. The ancient ancestors of these things look probably different than any of these viruses.
But I think if you look at, the other NCLDVs are just not as well studied, but I'm sure they're going to have their unique sets of genes as well, right?
Absolutely. So what I'm going forward with this, as I say, this has kind of awakened me, and I don't know if this will last, but I've already written Matthias and invited myself to the next meeting. I want to look at this from the other side and take the core genes, these things that are called core genes, and look at what they do in the pox viruses, so that we can look and see what of these biological functions are actually conserved throughout the NCLDVs. It's interesting because during the TWiEVO, when I brought this up about a conserved bunch of core genes, there was on the panel amongst our guests some skepticism voiced, saying, "Well, yeah, okay, conserved core genes. Well, you've got genes that might be conserved in a fungus, right? Does that mean you're really the same as a fungus?" But I want to look into this further, because as whether these core genes really distinguish the NCLDVs from other phylogenies. I think they do. And in fact, just thinking about some of the core genes that I know something about, some of the ones involved in regulation of transcription, for example. I can see where a DNA polymerase or a capsid protein or an RNA polymerase subunit might be conserved not only in the NCLDVs, but a lot of homology outside that as well. But some of these others really are pretty specialized for viruses that might have part of their replication cycle in the cytoplasm, like these late transcription factors and et cetera. So I'm going to look into this further, and I'm going to be corresponding with Eugene and Natalia in the process to make sure I get it right.
Yeah, that's cool. No, I think it's a good idea. And actually, Mart Rupovic kind of, I think, echoed or extended a little bit this idea. So you have these core shared genes based on at least some reliable sequence identity between them, which implies sort of a monophyletic origin or a single origin somehow. Although when you talk about giant viruses, I think for me at least, the take-home messages is that you can really scramble the deck genomically in a lot of different ways. And so then he's starting to look at things like shared genes, but in diverged viruses, and pointing out at least evidence from the sequences of really diverged functions. So for example, it might be a shared gene that has a known enzymatic function, but even just doing sequence gazing, you can see that the key amino acids for actually conducting the enzymology that's known from experimental biology or from biochemistry are totally different, and probably it's totally disabled. And so it's still just a lot of sequence gazing, but the implication is that you share that gene and you share the protein, but those proteins are doing different jobs because one is probably the kind of known biochemical function, whereas in some of these giant viruses, who knows what they're doing with it, because we're just looking at the sequences now.
Well, and there's a specific example of that in the stuff that I talked about as well, because my absolute favorite gene is this thing called A18, which is one of the most conserved core genes, and it just happens to be one of the things that I spent most of my career working on, and it has, by sequence gazing, has motifs that make it look like a helicase. And we looked at this in some detail and figured out that, in fact, in pox viruses, this is a transcription termination factor. And the helicase function, although we were never able to actually prove this, I presume, based on some comparisons in other systems, really has to do with the notion that this thing binds DNA and in an ATP energy-dependent fashion moves along the DNA and dislodges things in its way. And that brings about termination of transcription. And this thing is conserved throughout the NCLDVs, but does that mean it's really doing that kind of thing in these other viruses? Could be it's doing something entirely different.
Sure.
Yeah.
Totally.
And I actually, in my correspondence with Natalia and Eugene, asked that very question, and they asked it back to me.
Mm-hmm.
So
I would almost propose like kind of reverse conciliation here. So if we've been talking about cutting the pox viruses loose, and I think there could be some very good reasons for doing that. For example, if you want a stronger phylogenetic signal, I think you don't want to make assumptions about functions of core genes, since it's clear, just as you're saying, that these things can kind of go off in different directions. So then the question is, well, what if we start at sort of a shared function between the viruses? And so that's, this idea that they're gene thieves and steal genes pretty regularly from their hosts or gain them by horizontal transfer. And then if maybe there are sort of common themes in that process across, even if you do that in different ways, but sort of maybe concepts or principles that might emerge about the biology underlying that. And so that's sort of my sales pitch, I guess, for the reason for including pox viruses, is that we have all these experimental tools. We see, when we just look at the genome, sort of similar themes emerging of gathering genes from hosts or undergoing gene copy number amplifications, all of these things. And so maybe there is reason to believe, just in some of the mechanisms or concepts of how these viruses divide in diverse, crazy different hosts all over the world, that there are some shared features that make it worthwhile to include all as we consider this. And that's one of the things I loved about the meeting, was having those kind of late night conversations about, oh yeah, we just saw this, and I'm thinking, wow, I just saw that too, and kind of having those little sparks of connections between the different systems.
That's why keeping people in one place is great, because if you're at a meeting in a city at night, people run off.
Yeah,
they're not captive. At this place, even though the session ended right before dinner, after dinner, people have to stay there. And the cool thing, this place had a lot of rooms, right?
Yeah. And
there was one room where they had drawers of wine and beer, and you signed your own out, and then you went and found someone to talk to. It just was great. It was really good.
What is it? The witch's room? It's this crazy room with tapestries and
yes.
It has like you feel like you're in your weird uncle's basement or something. It's like the perfect spot to hang out and discuss viruses.
Now, I want to pick up on something you mentioned earlier, just briefly.
Yeah.
So you talk about this idea that, we have model systems, but should we be developing them with some of these big viruses? It's hard. I don't know if it's worth it. However, you end up with 50% novel genes. You want to know what they're doing, right? That's the problem. So
right.
People want to develop a system to figure that out. Unfortunately, for each host, it's maybe slightly different, right?
Yeah.
And compounding the issue is that funding for this is hard to get because it's not health related, right? So the sources are very hard. I mean, they exist, but much harder to get substantial money. So
yeah, there's a lot of these viruses where the discovery has to do with really basically just sequence. And for example, the choanovirus, you can't culture the host or the virus.
Yeah.
All right. So it's just a matter of sequence. There are a few systems, the AAV system, Steve Wilhelm's, and the E. Huxley virus, where you can culture both the host and the virus, and you know that they are a natural pair. And you can study the ecology of that pair, but also reproduce the biology in the laboratory. And those, if I were starting on this, I would zero in on one of those and try and do a lot of the basic biology in the laboratory.
Yeah, I think having cultured hosts is important, but still, they don't have plasmids, they don't have CRISPR-Cas work.
Right.
You still have to work it out,
right. Yeah. So now I would add like coronavirus to that mix since you can do the plaques.
Yes. Yes.
Absolutely.
Oh, yeah.
Yeah. And so I think it's really important to think through what are the tools that exist. Can you do plaques? Can you do some of the basic virology, which like that's a game changer right there. And then the next question becomes the one that you're raising is then can you start to manipulate the virus with genetic tools like CRISPR, et cetera? That can be a really tough slog, but making those investments is worth it. And it might kind of be an echo of the last generation of science, which is why do we today think of Mus musculus, the mouse, and Drosophila, the fruit fly, and C. elegans, the worm, and yeast, Saccharomyces cerevisiae, as the model systems. There was, in a sense, almost a evolutionary process where there were many more model organisms that were sort of run up the hole, sort of failed because you couldn't grow them reliably in the lab, or you didn't have plasma, it was hard to get the molecular biology tools to it. And so I think that kind of process is unfolding again, kind of in this generation of as we glimpse it all and go back to the oceans and all of this complexity of the ecology what is the prospects for picking out a few systems or what will become the model systems of sort of tomorrow. And so, and how far and wide can we spread that as the tools get more powerful? And so that's kind of, I think, happening in real time right now and is sort of where the breakthroughs of the future will be.
And remember, Nels, the systems we have, the model systems had advocates, right? Who pushed for them, like Max Delbruck, right? For phage. And so this, maybe these individuals don't realize that, but they have to advocate beyond what they're interested in.
Yeah.
And that takes a different kind of person. It's not everyone that's going to do that.
Yeah. Yep. And so I think, an example we've talked about even recently on TWiEVO, Nicole King, and how she kind of pioneered anaphylagians. That's one of these success stories that is a real model. But, I think as we talked to her early in TWiEVO days, that was not a slam dunk. And she spoke candidly about how much of a slog that was, how committed over, a decade she was, how sequencing the genome and then sort of slowly building those tools. And that's still happening. So yeah, no, it's really fascinating. But, Vincent, speaking of the plaques, before, we're kind of recording a giant podcast for this giant viruses, which I think I don't want to. Yes, I don't want to leave this on the sidelines. So this mass spec plaques, maybe share that story because I think we were all kind of enthralled by this work at the meeting. So
this is a story we covered on TWiEVO, but just briefly. So Asaf Vardi, who is working in Israel. And his, I think it was a postdoc or a student, Guy Schleier.
Yeah, student, I think. Guy
Schleier, who talked on the last day. He talked about the mass spec plaque assay. Asaf did not, I guess, leaving it for a student. So this is taking the plaque assay, which I say is the greatest assay in all of biology, and which hasn't been improved on since 1900s because it didn't need to be improved on. And a plaque assay, as they say, captures everything that's going on. The virus initially infects a group of cells. It reproduces in them, it may kill them and move on, and you make concentric circles of expanding cells infected to eventually form a plaque. And they said, can we capture what's going on in this? And they developed a mass spec assay where they take a set of plaques and essentially transfer them to a filter and then run a mass spec over it and say just to step back for a minute for the audience. I was thinking we should have done this for the podcast. Mass spec is a way of what it does is measure molecular weights of individual species in a sample, and it turns out that the molecules are so complex that they have unique molecular weights. So if you just compare simply a molecular weight with a database, you can get a good idea of what a molecule is. And this has to do with, how do you describe this? Shooting this molecule essentially down a tube and asking how fast it goes around a corner. Yeah, so and that's a function of the molecular weight. Right, they break up large proteins into small bits, and then they can identify the small bits by these various ways. And so on these, that's what they've applied to these plaque assays. And the organism is Emiliana Emiliania huxleyi, which is a coccolithophore found in the ocean, makes these huge blooms that you can see from outer space. And the virus that infects it, and they can grow E-hux in the lab. They can do plaque assays. And they already know that the virus reprograms the cell in many ways, metabolically, lipidomically, and so forth. So they can take this plaque and ask, what's going on as far as the lipids go?
Yeah.
And they can image the original plaque. So you can see, say you look for a particular lipid, and this is what he did. One lipid will have concentric circles in the plaque, showing there's different things going on at different stages of infection. And they found, for example, that the cell is reprogrammed to make odd chain carbon fatty acids as opposed to even chain fatty acids. And so this is an amazingly powerful approach. So you can start to say what's going on in the cell, and then, of course, you could go back and look at the biochemical pathways and try and modify them and see if, in fact, it makes a difference for infection or not. But I think this has huge implications for all of virology, because you don't have to just study E-hux. You can do any of your viruses. You could do it with a pox plaque. You could do mass spec on it and say, what are you interested? You're interested in the lipids? Are you interested in protein changes, other small molecules? You could query anything, essentially, in the plaque.
Yep. No, I agree. And it kind of gives this geography of the infection in a sense. So
the geography of the infection. Beautiful.
Yeah, because you start in the middle, presumably with a single particle that's then dividing and spreading the infection in those concentric rings. And why is it that the lipids are different, kind of at different times during those replication cycles? And you now, because you have that spatial dimension to it, you can kind of see that biology for the first time. And I also thought they were doing some really cool ideas talking to Guy at his poster with thinking about in that plaque or surrounding it, who are the cells that are actually resistant to the virus. So it's kind of
right.
single cell level. And because they've developed these tools or these ways of looking at higher resolution and in, applying these kind of classic techniques in a new way to the virology, you can start to think about, what are the populations of the cells and how are they reacting sort of differently, and what is the genetic basis of that as you do single cell genomics, for example. And so, I was blown away. I think what's going on. And so Asaf Vardi's lab over at the Weizmann, I think they're just doing really creative, cool stuff kind of on the front lines, both with giant viruses, but also, as you're saying, Vincent, with just kind of that could be applied to virology in general.
Yeah, it's great. So it's just amazing. And we were lucky to have him on TWiEVO and Alex and Steve. It was a great trio. Just amazing. Why don't we do a couple of rapid fire picks before we go? I mean, not picks, but what do we like?
Yeah,
I just have one more I want to do that blew my mind the whole time. Jeffrey Blanchard from UMass just went digging in the dirt, at some site outside UMass, and did both metagenomic sequencing and electron microscopy to see what was there in terms of giant viruses. And, when these first happened, they were happening at odd sites around the world, mostly aquatic environments of one sort or another. And you got the impression, I think mostly because of the history, that they were pretty rare events. And here Jeffrey just goes outside and digs up a bunch of dirt, and there's a whole bunch of new viruses that nobody's ever seen before, including a bunch of different morphotypes, right? The EMs were fantastic,
cool.
All different kinds of stuff. So these things are everywhere.
Oh, yeah. Yeah, and this is this kind of cool thing. So it's this site in the Harvard Woods or Harvard Forest in Massachusetts, where they've for years held little patches of soil a couple of degrees C higher than the rest to see what temperature is in temperature. And so that's where they're sampling the dirt to find these new crazy. Yeah, that was really fun.
How about you, Nels?
Yeah, so my last one is, and we've already mentioned Eugene Koonin and Natalia Utin a little bit, but so one of the fun things from their talks, both of their talks that came out of it, was I think kind of scrambling the notion of like what a giant virus is. So you've got now on the kind of coming that will be published soon, some new genomes that sort of put take the giant viruses down a few notches, and then take the smaller pox viruses and bring them up a few notches. So a lot of the giant viruses, people have been looking at are trying to kind of find who are the biggest ones. I don't know. There's this sort of human idea that bigger is better, that you can kind of almost unavoidable. But some of the mimiviruses and others are now weighing in actually at pretty modest genome sizes, something like 70 kilobases. So now you have to think, okay, wow, these aren't just these anchors. There are also these really kind of modest, quote unquote, giant viruses replicating all over the place. At the same time, and for kind of selfish personal interests, there's a pox virus that Eugene was telling us about, carpedema virus, that looks like it's going to be just under 500 kb, sort of twice the size of cowpox, which up until now is sort of considered one of the large pox viruses. And so I think, just the sort of simplistic idea of using genome size is sort of out the window, because as we get a better handle on the genetic diversity there, it's going to be a pretty wide range sort of across the board. Mm-hmm. The other point, just sort of corollary on this, that Natalia brought up was, so most of the viruses, the giant viruses we've been discussing, are all in single cell eukaryotes, a lot of protists, algae, etc. She was describing to us these mimiviruses, so in a class that's known, but what's important about this is the host is different. In fact, it's an animal. It's not just I shouldn't say just, but it's not a single cell eukaryote. It's fish. And so there's a new collection of sturgeon viruses and other mimiviruses. And this is actually kind of important because it kind of, again, muddies the waters a little bit, so to speak, about what is the host range of these things. Right. And now kind of, the more simplistic view of, okay, pox viruses hit the mammals, and they are smaller, et cetera. Those are, I think you can throw the rule book out the window, and we're still just discovering all of this diversity. So I'll close on one idea, which is a little more, so taking and using Natalia. So she, of course, this is an international meeting with folks from all over the world, and so it was conducted in English. But then, giant virus meeting after hours turned out to be a really fun musical open mic scenario. So we had Japanese colleagues singing songs. We had Matthias Fischer put together an entire virus opera that was just sort of over the top. And then Natalia actually sang some Russian folk songs that I thought was just really striking, how incredible and professional she was, professionally communicating through music. I wish she had her guitar actually when she was giving her talk about viruses. It was just so almost poetic. And I think it also illustrates or maybe echoes on your point, Vincent, that, having a small meeting like that you just sort of is a great way of building community beyond just the science, but also to have some fun, to sing some songs together, to draw, as we did, draw some giant viruses and some coronaviruses using, it was just really, what a positive, fun way to conduct science.
This little session showed that scientists can be creative, right? I mean, Matthias is a terrific piano player. There was another guy at the meeting who joined later, was playing the piano, and they're talented. They don't just do science. It's really cool. This sturgeon mimivirus was published in 2013, and we missed it because it was published in Diseases of Aquatic Organisms.
Right.
Yeah, this is a pathogen, right? It kills these fish
apparently,
dead in large numbers.
It's called Namau virus. They found it in sick sturgeons. It looks like, and they do EM on the tissue. They can see the particles there. It looks pretty good. I mean, they admit that in the paper we still have more to do to prove that this is causing the disease. But and I would say if it's in fish, it's going to be in bears and maybe even humans. That's one of the open questions that was put on the table. I'm really excited about the notion of more fish viruses. We're getting more and more interested in my lab in zebrafish, and so the more viruses, the better. It would be fun to have some more virus host systems in a kind of old school genetic system with experimental. So let me end up with one more, and I think we have to mention James Van Etten.
Yeah,
because he started this field unknowingly many, many years ago with his virus of the algae that are inside of Paramecium, PCBP1. At the time, it was the biggest genome known. And he's at University of Nebraska. He's now in his 80s, right?
Yeah.
Comes to all these meetings, travels all over the place. He persisted. He kept studying this virus. It makes great plaques on green lawns, which are fabulous. But at this meeting, he's asking questions. He's so excited. He's so into everything that I think it's remarkable that he can be working for so many years and still have that kind of passion and enthusiasm. And, he does stuff that is really not related to human health, but nevertheless sets really interesting precedents. So he talked about, when he infects algae with his virus, within 7 minutes, the DNA is in the nucleus and it's being transcribed. Holy crap. Even though it's giant, it's like a phage.
Yeah.
Right.
They encode the complete machinery that glycosylate their proteins, including the major capsid protein. Mm-hmm. The whole machinery to do this is encoded, and he's really interested in these enzymes that attach sugars to viral proteins and what they're doing and so forth. So he's really pushing the sugar chemistry with collaborators in Italy, and it's just a fraction of what he does. But I think, hats off to him for continuing when he was the only one. And now he's got all these other people. He just loves it. He's like a pig in shit.
Yeah. The PBCV was discovered in 1981. Okay. So long time ago. And Jim showed up at some of the first poxvirus workshops because, he had this thing that didn't fit anywhere. Yeah. And the closest fit was poxviruses. So he used to come to our meetings, but at the time was sort of, out on the fringe. Though we were thrilled to have him because he's such an engaging individual, and the system is so interesting. And now he's sort of, the linchpin of this whole thing.
Well, I think it amplifies nicely on your point too, Rich, which is that for kind of as new fields are kind of emerging in the early days, the early meetings with small groups of people, that the kind of personalities at the base of the tree, so to speak, really matter. And so having kind of a generous, creative, curious person like Jim Van Etten, that can kind of set the path in a really positive way. And so I think it bodes well for an emerging field to be optimistic about all of the prospects of incredible biology coming together, which just sort of captures the imagination of structural biologists, ocean ecologists, virologists, and evolutionary biologists. That's a pretty cool thing. And if it kind of gets off on a positive foot with folks like this kind of showing the way, there could be some real headwinds and challenges in terms of, well, what is the very strict medical, kind of application from a funding perspective? But I think from a biological perspective, there's just so many incredible things that will sort of emerge from this that we can't even imagine. And so, I think keep an eye on giant viruses. And if any of our listeners are kind of looking for cool systems or thinking about ideas of things to tackle, I think you would do well to point yourself towards this increasing sort of swath of really incredible biology.
And he has influenced people because there were a number of talks and posters on PCBP there, right? Mm-hmm. There's structure, as we talked about, but also one guy doing evolution of the virus and its host. So, I mean, he's just terrific. And, of course, he and his wife always wearing red.
Right.
Cornhuskers showing up at the German castle.
Hey, let's wrap up with some picks. Rich, what do you have?
So I can't believe I haven't picked this for TWiEVO, but it doesn't matter because this is TWiEVO. This is an app called Flight Radar 24 that I showed to Vincent on our way back, and this is real-time data on all of the airplanes in flight at any given time.
Oh, wow.
Globally. So the most amazing thing about this is that if you just boot the app without picking a particular flight and zoom out, it looks like, ants on some delicious morsel of food or something like that. There are, of course, thousands of airplanes in flight at any given time. And if you zoom out, you can sort of see the flight paths and that kind of stuff. But you can also pick any given flight, like the one your friend happens to be on at a particular time, and it'll track them, tell you their altitude, their speed, where they're coming from, where they're going. It even has a picture of the plane, and I guess there are, if you click on the picture of the plane, you get into a photo library of the airplanes where they're, I guess people who are airplane geeks go around and track airplanes of, given individual airplanes with given individual registration numbers. So you can get pictures of an individual, that airplane in its various lifetimes. It may not have always been flying for that particular airline.
Yeah, it's so cool.
So this is everything you ever wanted to know about both individual flights and the global flight information in a very provocative application.
I think that the history of the aircraft is so cool. I didn't realize that. Yeah. Okay. When I took off, you were still on the ground, Rich. So you sent me a picture of my plane.
Yeah, I was tracking you.
That's funny.
So cool. This is just awesome. I love this.
Yeah, that's neat. Looking at the map now, it doesn't look like that big blizzard is slowing down air traffic across the western U.S. There's still plenty of planes in the air all over the place.
So we had an image in the textbook, virology textbook, in the emergence chapter to illustrate, the extent of air travel and the globe. It was an old image with lots of roots, but I took a screen capture of the whole world with all the little planes. It's much better. We're going to include that in the next edition.
Okay, great.
It's very cool. I love it. Nels, what do you have for us?
Yeah, so my pick is the Deutsches Museum in Munich. So when you guys were at the end of the meeting, as you guys were migrating back and getting on those flights, transatlantic flights, I ended up cooking in a day or two in Munich. So I've been kind of in and around Bavaria years ago, but I haven't spent any time in Munich, and so added a day, 2 days about to the itinerary, and used one day to visit the Deutsches Museum on this island right near downtown Munich. And this was on your recommendation, Vincent, and it was a good one. So the kind of masterpieces of science and technology, and just all of these, museum halls with on different topics. So like things like energy or electricity or air flight or underwater navigation, and then seeing kind of the history of that with museum pieces that sort of highlight some of the innovations over the centuries in each of these topics. One of the really cool things about this is a lot of the original U-boats, the submarines, the German submarines, are actually cut in half. So you're looking at an entire U-boat, but it's almost like you're immediately inside of it because it's in cross section, because it's been physically, with blowtorches or whatever, cut in half. And to kind of keep doing that and to just sort of see these, whether it's boats or submarines or airplanes, some of the original Wright brothers, brand right at the, emergence of airplane flight, to kind of see that laid out in front of you. It's really inspiring and kind of brings it to life in a different way that I think is really unique to that museum. So anyway, I had a blast spending an afternoon there, and then going to some beer halls in the evening before I flew back to Salt Lake from the giant virus meeting.
Good. I'm glad you liked it. It's a great museum.
Well, I need to do this. I actually visited that museum in 1963.
Wow.
When I was 15 years old. And I remember very vividly how hands-on it was.
Yeah.
It was a terrific interactive experience. And you've really got me stoked here. I want to go back.
It's worth it.
Hey, in 2 years, you go to the giant virus meeting. You can check.
Yeah.
Yeah, good point.
I'll go with you.
How about you, Vincent? What's your pick of the week?
So I have a site called Diversify Microbiology. There are actually a bunch of these. There's Diversify Immunology. I think Kathy Spindler passed on Diversify Immunology. It is a collection of people, basically, who are from traditionally underrepresented groups. So this one's microbiology. If you're organizing a meeting, you want women, you want racial or ethnic minorities, people with disabilities, LGBTQIA. You can find them here. And these are, people can nominate other people to be on this list, or they can put themselves on. And I think it's good because, people typically think of the same people over and over for meetings or whatever, and this can help you diversify, which is cool. There's also a link to what they call resources, and they have other things that are useful in the kind of the same vein, women speakers at meetings, minority microbiologists, women virologists, diversify chemistry, diversify immunology, and so forth. So you want to think a little bit differently, as you should. I think this is a useful resource to have.
Very that's interesting.
Yep, that's great. And actually, that would be kind of my, hope or wish for the giant virus community, that more and more diverse voices with different backgrounds, folks from all over the place, that it will just keep growing in its diversity to almost mirror, in some sense, the diversity of all those viruses that we're just glimpsing at. That would bode really well for sort of the vibrancy of the movement. Sure. Yeah.
All right, that is a giant TWiEVO on giant viruses. I got 2 hours coming up here.
I was wondering, as we were going into this, how are we going to fill up the time?
It's never a problem.
Never a problem.
Here's the thing, Rich and Nels. When we get together, we talk all the time, right?
Yeah.
It's never an issue. There's never a silent moment.
Right.
Yeah.
And so, because we're interested in stuff, and a meeting like this just stimulates, your thoughts about the topic. So we never have trouble filling time. We could sit down with no agenda.
Right.
And talk for 2 hours.
Right.
Guilty as charged.
Microbe.tv/TWiEVO, where you'll find show notes, and I'm going to put a bunch of pictures up there, so you should go and check them out. TWiEVO at microbe.tv. You can send us your questions and comments, pics, whatever you'd like to do. If you listen on a phone or tablet on an app, please subscribe so you get every episode, and we know how many people are listening. It helps us to raise some funds. And speaking of funds, if you like what we do, consider supporting us financially. Everyone does this on their own dimes and could use some help to pay our expenses. Microbe.tv/contribute. Rich Condit's a emeritus professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thanks for joining us, Rich.
Sure thing. What a blast. This is great. Thanks, guys.
Yeah, the meeting was great, and I enjoyed this as well.
Yeah. Here's to more. More of the same.
Yeah, I agree. Nels Elde's at cellvolution.org over on Twitter. He's L. Earlybird. Thanks, Nels.
Yeah, my pleasure. And just to say, so I think in our giant 2-hour podcast here, I've got 2 inches of snow that came down.
Wow.
Oh.
In the middle of it. It's
cool.
Really fun.
So you can see it right out your window there?
Peeking out the window of Elde Lab Studios and watching winter arrive here in real time. But yeah, no, this was really fun. And I'm looking through my meeting notes and just thinking that oftentimes we do these meetings, it's really intense few days, but then it kind of fades really quickly. It's really fun actually to do this recap, and it kind of brings it back to life. And I think it'll have more of an impact on my thinking in the coming months, the fact that we sat down and did this. So thank you both. That was really fun.
Yeah, it's kind of like a debriefing where this is of course a record. It'll always be out there for other people as well, but it helps to go over your notes and think about it again for sure. Yeah. So thanks for agreeing to do this. I'm Vincent Racaniello. You can find me at virology.ws. Music on TWiEVOs is by Trampled by Turtles. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Till then, be curious.